Okay, today we will talk about urea cycle. Okay, for today learning outcomes, you will get to know the importance of urea cycle, understand each process that involved in urea cycle, and last but not least, get to know the overall reaction of the urea cycle.
Now, the importance of urea cycle. The first thing is it can excrete the ammonia. After the metabolic activities, our body produces metabolic waste including ammonia, which is very highly toxic and needs to be converted to other forms to reduce the toxicity. Hence, it is converted to urea by going through the urea cycle. Number 2 is urea cycle can help produce fumarate. Fumarate is the side product of urea cycle. It will act as a bridge to link urea cycle and gluconeogenesis. Gluconeogenesis is the process to produce glucose from fumarate. And the third is maintaining the pH. As we all know, carbon dioxide is a substrate in urea cycle in the form of bicarbonate. The usage of the CO2 in urea cycle will help in maintaining the level of carbon dioxide.
So we can see this diagram that show briefly about the urea cycle. For the urea cycle, we have five process, which is the first process is to form the carbamoyl phosphate, and then it combine with the ornithine, and then it will produce a citrulline. After that, the citrulline will combine with aspartate that form argininosuccinate, and then it will be again do the process and form the arginine. That's after that the H2O combine with the arginine, and then you will excrete the urea, and then it will return to the origin, which is ornithine. And then we can see this is the formation of the urea. The first C is get from the first step, which is carbamoyl phosphate, and then this NH2 is from the ammonia itself, and then NH2 it gain from the aspartate, and O is from the H2O.
Before we start the urea cycle, we will briefly explain about the transamination of amino acid and also oxidative deamination.
Transamination. Transamination provides ammonia and aspartate, which is then used in the urea cycle. As we can see here in the urea cycle, the aspartate is derived from the transaminations of oxaloacetate, which is being catalyzed by an enzyme known as aspartate aminotransferase AST. The AST transfers the amino group of the glutamate to the oxaloacetate, forming aspartate and alpha-ketoglutarate.
Ammonia in the urea cycle can be produced through the oxidative deamination. The oxidative deamination occurring in cells involves the amino acid glutamate, which can be oxidatively deaminated by the enzyme glutamate dehydrogenase. This reaction is either using NAD+ or NADP+ as a coenzyme. This reaction generates alpha-ketoglutarate and ammonia. The free ammonia will be used in urea cycle.
Carbometyl phosphate synthesis 1, CPS1. Step 1. Formation of carbometyl phosphate. Carbometyl phosphate is formed by carbometyl phosphate synthesis 1, CPS1. CPS1 is a ligase enzyme located in the mitochondria involved in the production of urea, which requires an acetyl glutamate to become activated. CPS1 transferred an ammonia molecule from glutamate to a molecule bicarbonate that has been phosphorylated by one molecule of ATP.
This reaction takes place inside the matrix of mitochondria in the liver cell. Carbon dioxide in our body exists in the form of bicarbonate. Bicarbonate is phosphorylated to become carboxyphosphate, and it is required 1 ATP. Carboxyphosphate will later react with ammonia, which is provided primarily by the oxidative deamination of glutamate by mitochondria glutamate dehydrogenase. And then it will produce carbamate. Carbamate is phosphorylated to give carbamate phosphate, which requires 1 ATP.
The molecule that have formed is high molecule that has a high transfer potential known as carbamate phosphate.
Next is the formation of citrulline. This formation takes place in the mitochondrial matrix. Carbamate phosphate has higher energy and is ready to react with ornithine. Carbamyl phosphate and ornithine are synthesized by ornithine transcarbamylase. This reaction is activated by N-acetyl glutamate. Carbamyl group then kicks off phosphate and combines with ornithine to form citrulline. Citrulline is then released into the cytosol by transporter system.
The synthesis of argininosuccinate. This process occurs in cytosol. The first amino group in urea came from the ammonium. Meanwhile, the second amino group in urea came from aspartate. Aspartate combined with citrulline by the help of argininosuccinate synthetase. The formation of argininosuccinate is driven by the cleavage of ATP to adenosine monophosphate and pyrophosphate. In the formation of urea, this is the final or third ATP molecule used.
Okay, now let's move on to the next step of urea cycle, which is the cleavage of argininosuccinate. In this step, argininosuccinate is cleaved to yield fumarate and arginine by argininosuccinase or arginosuccinate lyase.
The fumarate that is produced is hydrated to malate, providing a link with several metabolic pathways. For example, the malate can get oxidized to oxaloacetate, which can be used for gluconeogenesis. Alternatively, the process can continue with conversion of oxaloacetate to aspartate via transamination, and the aspartate can re-enter the urea cycle.
Now, here comes the last step, which is the cleavage of arginine to form ornithine and urea. So the enzyme involved in this step is arginase. So arginase will hydrolyze arginine to form ornithine and urea. So in these steps, it involves one molecule of water, as it is hydrolysis process. The final product in this step, which is urea, will then diffuse from the liver into the bloodstream and finally will be transported into the kidney, where it will be filtered and excreted in the urine. So basically, in this step, we want to remove these two amino groups from arginine. When these two amino group is removed and then combined with an atom of oxygen from water, it will form urea. And this urea will diffuse and excreted in the urine. So that's all. Thank you.